In this video, we're going to be learning about B lymphocytes. Now, before we continue, let's put this into context. So step 1, we had our physical and chemical barriers. This was then followed by step 2, where white blood cells, such as macrophages and neutrophils, engulfed and digest pathogens. They turned into APCs. The APCs then activated T lymphocytes, which then killed more infected cells, activated more APCs, and now they're going to activate B cells. The B cells will make antibodies, and this should hopefully clear the infection. Okay, so a B lymphocyte is a type of white blood cell. However, on its surface, we have a special protein called an antibody. Now, in the body, we have millions of different B lymphocytes, and what makes them unique is that they all have different shaped antibodies on their surface. These are called B lymphocyte clones. Now, the B lymphocytes response is also known as a humoral response. The humor is an old-fashioned word for blood. That means the B lymphocytes will be functioning mainly in the blood. So, the B lymphocytes travel in the blood and eventually they're going to come across some antigens. Now, these antigens can be floating freely or they could be on the surface of a pathogen. In any case, the B lymphocytes keep moving towards the antigen. Now notice here that these B lymphocytes have different antibodies on their surface. Only the B lymphocyte with the correct antibody will be able to bind to the antigen. So we can see that it was this B lymphocyte that had the complementary antibody. This process, where the correct B lymphocyte clone is selected, is called clonal selection. This B lymphocyte will keep going until it finds a T helper cell. The T helper cell will be able to bind to it because it has a complementary receptor. It will then stimulate the B cell. Once stimulated, the B cell will then divide by mitosis to make many copies of itself. These copies turn into B memory cells and plasma cells. B memory cells stay in the blood for a long time, whereas plasma cells immediately start producing antibodies. Very soon, these plasma cells disappear. However, if the same antigen or pathogen reappears, the B memory cells will differentiate into plasma cells. And once again, we can produce lots of antibodies. Okay, so to summarize. Number 1, there are many clones of B lymphocyte in the blood. These clones have different antibodies on their surface. The antigen binds to the complementary antibody on the B cell. This process is called clonal selection. Step 3. A T helper cell with a complementary receptor will bind to the B cell. The T helper cell stimulates the B cell to divide by mitosis. As you can see, now we have many copies of the same B cell. The B cell creates copies which differentiate into plasma cells and B memory cells. Plasma cells produce lots of antibodies in a short period of time, whereas B memory cells remain in the blood for a long time and can quickly turn into plasma cells if the body is reinfected with the same antigen. Hey guys, if that video helped you, support our channel by liking, subscribing and sharing it with your friends. And more importantly, if you still have questions, drop a post on our forum at ExamQA.com, where I will personally be there to help answer your questions. Mohammed signing out.